This conference will now be recorded.
Hello everyone. Chemistry in everyday life. This is a new chapter we are starting today. This is the first class. So as introduction we can say in our day-to-day life, what is the role of chemistry? It plays a vital role in our daily life. And if you consider the three basic requirements without which we cannot survive, that is food, clothes and shelter. So in all these things you will find application of chemistry, especially if you consider clothes. In the last chapter that we have finished, polymer. So in cloth related things, the application of polymer is really significant. Fine. So that we have also seen. Now other two factors are food and shelter. Okay. Now medicine, that is also important for survival purpose. So it is also uh, which is used to treat illness, that is also made of some chemical compounds. Fine. So in fact life itself is a complicated system of different interrelated chemical processes. So whatever is reactions going on inside the body, that is in vivo, sometimes even we do not know the details of all these reactions, but just because all these reactions are going on naturally, so that is why we are alive, right? So it is a very complicated system, but we'll focus mainly three fields in this chapter, which is medicine, then food materials and cleansing agents. So these will be the three main field, because there are so many fields. You can see in this picture also there are so many fields, but we will focus only on these three. That is all about this chapter. Fine. Here we have some examples that you can see in day-to-day life: combustion, rusting, digestion process, which is going on in vivo. Then photosynthesis, which is occurring in plants, which is basically giving us the energy source. Then battery electrochemical reactions are going on, fermentation, for example, wine. Then washing hands, which will be discussed in this chapter only, because cleansing agents are there, and also the baking process. Okay, so in all these things, just I will take one or two examples. Suppose if it is combustion, fine. So it is required if we want to make food. So for in combustion, what happens? It is generally the hydrocarbon compound in presence of oxygen, this reaction occurs. So it is reaction between oxygen and hydrocarbon compound, and products are carbon dioxide and water, and lots of energy, and this energy helps us to prepare food. Not just for prepare food, combustion is also you will find that is in how the vehicles are running. So all these, in all these things there is role of combustion reaction. Then rusting process, pure iron, if it is in presence of some impurity, and obviously in presence of oxygen and moisture, rusting process occurs where iron, that is pure iron, now it will be converted to some its oxide. And in digestion, in presence of hydrochloric acid, our food it is disintegrated in small particles, and after all, though this process is not so simple, I'm just saying what happens in stomach. So it is mainly the presence of some hydrochloric acid and also other enzymes. Fine. So it helps in digestion of the food. So these are some of the examples, but mainly we'll focus on medicine, food, uh, materials and cleansing agents. So first it is we'll see these three different areas. The first one is use of chemistry in medicine. This is the number one. Medicines or drugs. So when I'm saying medicine or drugs, basically drug is more general term. When I'm saying drug, it is not that all the drug molecules are medicine. Fine. So only some molecules some molecules of drug we can call it medicine. So whether it is medicine or drug, they are basically chemical substances. They alter either the physical or psychological functions of the body. Now this change, it may be for our benefit or it may not be. So when it is for our well-being, then only you can say it is medicine, or it is legal drug molecules. Fine. So they interact with the macromolecular target that are present in our body, and they produce suitable biological response. So I'm talking about in the second point only about medicine, because not all the drug molecules are medicine, though mostly we use you will also see that we are using the term drug in place of medicine also. So when we are using the term drug, it is mainly used for the replacement of the word medicine. Okay, so they interact with the macromolecular target in the body, and there is some suitable, that means some desired biological responses. So in this way, treat us when we fall ill or if there is some injury. Okay. Now the second area is chemistry in food material. So different chemicals are added to food, and these may be for different reasons. Okay, so these chemicals as food additives, we can have these 6 main categories. Food colors, sometimes we have to add some food colors to enhance its appearance, and then flavors, sweeteners, I mean artificial sweetener, then food stabilizing agent so that its lifetime is increased, antioxidant, food preservative to increase its longevity, nutritional supplement. So though there are 6 categories, but only 3 categories will be discussed in detail. And the last area is chemistry in cleansing agents. So we bath and wash our clothes daily by using different types of chemical compounds, whether it is in the form of soap, detergent, and any other cleansing agents. But if we consider the basic property of all the cleansing agent is that ability to extract the grease or dirt, okay, which are mainly organic molecules. It may be hydrocarbon also, okay, and as a result clean the surface of the substance on which they are applied. So in detail all these 3 areas will be discussed. The first area that we are going to focus is number 1, which is use of chemistry in the field of medicine. So first we will focus on this term, which is drug. So it is a very widely used term. So only some drug molecules are medicine, fine. But in general, if we want to give the definition, it is basically a chemical substance that can affect or alter the physiology when it is inside the living system. And these drug molecules, it may be naturally present, or it may be synthetic. That is, we can also chemically synthesize them in laboratory. It is used for the purpose of diagnosis, prevention, cure or relief of a disease. So not just for cure or prevention, for diagnosis also it can be used. Now, when a drug molecule is therapeutically active, that means out of different drug molecule is something which can affect or alter physiology. But when it is used for some therapeutic value, it is having some therapeutic value, that is for diagnosis, treatment or prevention of a disease, then only you can call them medicine or legal drugs, obviously. Now an ideal drug. Now this term that we are using in the last point, that is basically another way of saying medicine. So you can, though we shouldn't call it drug because drug may have different types of meaning, but mostly it is the commonly used term. So that is why we are saying an ideal drug is the one which is non-toxic. So it should not have non-toxic. Suppose it is therapeutically active, but simultaneously it is also toxic, then it will be of no value. It has to be non-toxic also, beside being therapeutically active. Then biocompatibility. Obviously, it should be compatible with biology, okay? And then biodegradable. It should be naturally degradable, and it should not have any side effects. So if all these criteria are fulfilled, then only we can take them. That is, it is possible to take in vivo inside the body. It can be entered. So that is the reason these conditions must be fulfilled beside its therapeutic value. Generally, most of the drug molecules that are used nowadays have the mentioned properties. Which mentioned properties? The previously mentioned properties, all these, okay? And they are at lower concentration. It has to be at lower concentration because we cannot take it at a higher concentration, because sometimes what happens when you are taking it a higher concentration, there may be some side effect. Toxicity may be there. So these conditions, mainly the conditions that are mentioned in the last point in the previous box, This condition must be fulfilled at lower concentration, because mostly what happens when it is present in higher concentration, suppose it is having a therapeutic value in higher concentration, then we cannot take it as medicine, because though it is having therapeutic value, but if you are taking in higher amount, there will be some other side effect, there will be some toxicity. So these conditions must be fulfilled, and also uh, at lower concentration, then only we can take it. And because at higher concentration there will be some other problems. Medicinal value of a drug we can measure by this term. See what it means? Therapeutic index. So if therapeutic index value is very high, that means its safety level is also high. So what is this index? How we will measure it? It is actually a ratio. It is a ratio between maximum tolerated dose of a drug. That means if you cross this value, it will be having some toxicity. So you cannot cross this value. That is the maximum amount you can take. So this will be in numerator, and in the denominator, it is minimum curative dose. That means if you are taking lesser than this amount, then it will not have any value. So you have to take this minimum amount. Okay. So in the denominator, you have the minimum amount that you have to take, and in the numerator, you have the maximum. You cannot cross this value. That means if this index is very high, this ratio is very high, it means what? That means minimum is the value, better it is. So when denominator is becoming low and low, obviously the ratio will be high. And when numerator value it is very high, that means the index is also becoming high. So maximum tolerated dose, if the value is very high, then also this index will be high. So that is why it is mentioned here that higher is the value of this ratio or index. That means safety range is also very high. Safer is the drug. So that is why if we know the therapeutic index, suppose 3 to 4 medicines are given, they are for the same purpose, but each one is having different therapeutic index. So from the therapeutic index, you can understand which one will be maximum effective. Okay. Classification of drug. We can do this classification based on different things. Though out of this different classification, mostly we will focus on the last one. But let's see what is this one by one. On the basis of pharmacological effect, what type of effect they are actually bringing about. On the basis of drug action, that means how they are doing this effect. That is, what is the actual mechanism. Now the third point is on the basis of chemical structure. That is, there will be similarity in the chemical structure. So suppose we have total 10 drugs. Out of these 10, 2 are having similar type of chemical structure. So they will be in particular group. So it is based on chemical structure. And the last one, which is the most important, is on the basis of molecular target. Because drug molecules, they interact with some specific target molecule. So depending on what type of target molecule they are interacting with, depending on that, we can have this classification. So one by one, we will see. The first one, which is based on pharmacological effect. This is number one classification. Different way you can do the classification. So how a drug or medicine is affecting or influencing the cell of an organism. That is actually pharmacological effect. So what type of effect it is bringing? In this classification, they are grouped based on the effect, on this particular effect that they are producing on the recipient, that is to whom it is applied. So for example, the medicine that have the ability to kill pathogenic bacteria, they will be grouped under antibiotic. Then another group is analgesic, which can reduce pain. So the first one, the first example that you are seeing, they are having some ability to kill the pathogenic bacteria, and they will be under the class antibiotic. Another class is those medicines which can reduce pain, they will be called as analgesic. Now this kind of grouping will provide the full range of drugs that can be used for a particular problem, condition or disease, whatever. So the physician, they have to carefully choose a suitable medicine from this available range. Okay, and that is based on clinical condition. So if the clinical condition is, that is our purpose is to reduce pain, obviously we have to give something from analgesic category, not antibiotic category. Okay, so from this whole range, here you can see seeing only two example, but there are other categories also. So based on from all these different available drug molecules, the physician, they have to choose carefully which one is actually required. The second one is based on drug action, how they are doing this action. Different drugs are acting differently, that is its own way of generating a response. So suppose two drug, they are actually doing the same effect. Suppose both are giving some relief from pain, but it may not be that both are operating in a similar way. Fine, so there may be some difference. It is based on drug action. It is more specified according to how it generates a response. So here you can see this example. Hypertension medicine, they are basically used to reduce high blood pressure. Now suppose we have two hypertension medicine, number X and Y. Now both X and Y, they are reducing this blood pressure, but how they are doing so, the way they are doing so, that is not same. Okay, so based on drug action, though the action is same, but how this action they are actually doing, based on that also classification is possible. This is number 2. Number 3, based on chemical structure. Now it is easily understandable. In this classification, drug with common skeleton. Okay, so common skeleton, when I'm saying, it means what? Suppose in a particular drug, there is presence of this heterocycle which is known as furan. Okay, and some other groups may be different. So this basic, though I am saying here it in a very simplistic way, it is not so simple because structures are not so simple. So common chemical skeleton, I am just mentioning, suppose this type of heterocycle is present, and now depending on what type of other groups or branching are there, based on that you can have different drug, but they are having some similarity in the basic structure. So they will be grouped under one particular category. Fine. So the drugs with a common chemical skeleton, they are classified into a single group. So here we have some example: ampicillin, amoxicillin, penicillin. All these are having similar structure. So see, as I have said, I am saying, giving this example, it is very simplistic way. So here these three molecules, you will also see their structure. When I'm saying that they are having similar structure, actually a large part of the molecule is actually similar structure. It is not only a particular heterocycle is same, so they will always be under same chemical structure category. It may not be so. Okay, so when it is mentioned that they are having similar structure, that means if you consider their total structure, a large part is actually same for all these three. Only some branching or a small group is different. So all these three names as it is given here, they are under the single group which is penicillin. Fine. So compounds, when they are having similar chemical structure, obviously we can expect that their chemical properties will be same. And if we talk about pharmacological activity, mostly it is same. Often have similar pharmacological activity. Just forget about this drug and all this discussion. If I'm saying that two compounds are having same type of chemical structure, that is 90% chemical structure of both these compounds are same. So you can expect that their chemical properties will be same, right? But when we are talking about drug molecules, as they are interacting inside our body in vivo, there is so many complex things, other complex things are present. So it is true that they will have similar chemical properties, but not necessary that their pharmacological activity will always be same. So that is what is mentioned. often have similar pharmacological activity, but not always. Fine. So for example, here two different examples you will see. All the drugs that are under penicillin group, it is observed that their biological action are actually same. That means in this case, their chemical structure same, their pharmacological activity is also same. But there is another two category. One category is barbiturate, another category is steroid. So under barbiturate, all the drug molecules that are present, though their chemical structure is same, which is based on barbituric acid, though their chemical structures are same, but they are not having same biological action. Similarly, steroid, it is not that, it is a general term. Under this particular category, we can have different drug molecules where chemical structure is same, but biological action is not same. So both are possible. Their chemical skeleton is same, but not necessarily their biological action will also be same. It may be same, it may not be same. And the last point, C, generally drugs that have same drug action and pharmacological effect. Suppose drug action, that is the previous point, the mechanism by which they are showing this action and the pharmacological effect. If it is found same for two drug molecules, suppose for two drug molecule X and Y, we have found they are having same drug action, their pharmacological effect is same. So in that case, you can predict, obviously they should have same basic skeletal structure with a minute variation in the branching or grouping. Okay, so when both drug action, that is a mechanism by which they are showing this property, and the pharmacological effect, that is what type of effect they are actually showing, whether they are reducing pain or they are reducing high temperature or they are reducing blood pressure, if that effect is also same, then you can expect that their skeleton structure must be same, obviously with a slight variation. So this is the third point, which is based on chemical structure. And the last one, which is the most important, and you will see mostly we'll extend our discussion on a drug, particularly based on this. So this is based on molecular target or site of action. That is where they are actually showing this action, site of action. Drug molecules, they target the macromolecules. Now by macromolecules, there may be different examples: carbohydrate, lipid, protein, nucleic acid, all these are present inside our body. And drug molecules, they can interact any of these macromolecules and they produce a biological response. So these macromolecules are basically target molecules for this drug molecule. So you can call them as if they are drug target. So the protein may be drug target, nucleic acid may be drug target, like this. And this classification is highly specific. That is the number 4th classification that we are discussing right now. This classification is highly specific if you compare with the other 3 classifications that we have already done. Okay, so that is, it is most important. These compounds often have a common mechanism of action as the target is the same. So suppose some drug molecules that are interacting with protein, a particular protein, because I am not saying any protein, obviously a particular protein. So target is same for two drug molecule. So the mechanism, you can expect that will also be same. Drugs that have the same mechanism of action will have the same target. So on the contrary, you can say the same fact, but in a reverse order. That also you can see. And it is the most useful classification, especially for physicians. Fine. So it helps them to choose the correct drug easily if it is already known on, that is, what is their target or site of action for these drug molecules. So the last point, now if we extend it, drug target interaction. The biological processes, for example, metabolism process, cell signal process, that is, some signal will be transferred from one cell to another. So all these, etc., all these things are very essential if our body is functioning normally. Okay, and these routine processes, day-to-day basis, all these processes are required. It may be disturbed by any external factors. It may be microorganism, that is virus, bacteria, um, fungi like this, or any chemicals, or maybe there is some disorder in our lifestyle, and that is why they direct effect on the system. So all because of all these processes, these biochemical processes, metabolism, cell signaling, they may be disturbed. So under such condition, we have to take medicine so that we can get back the original normal functioning of the body. And that is the reason the drug molecules, they interact with biomolecules such as protein, lipid, etc. that are responsible for that particular function which is disturbed. Here we have example, proteins which acts as biological catalyst. We know they are known as enzyme, right? Catalyst, definition of catalyst is which it is a particular compound which when added to a particular reaction, there is no change in its own chemical structure, but it enhances the rate of the reaction by decreasing the activation energy. The same definition, suppose this catalyst, if now it is found inside the body, biologic, now we are saying it biological catalyst, so mostly they are called enzyme. And enzyme molecules are mainly protein molecules. So biological catalysts, they are known as enzyme, and there are other molecules, that is those molecules which are important for communication purpose, they are known as receptor. Fine. So two different terms, one is enzyme, another one is receptor. Drug interacts with these receptor molecules. This means receptor. And modify the normal biochemical reactions. Now they can do so by two ways. Either by modifying the enzyme activity. So under normal conditions, suppose there is no drug, we have not taken any drug. Under normal condition, enzyme, they interact with the receptor. Okay. But if there is some disturbance because of some factors, now we are consuming this drug, not always consuming, it may be externally injection also. So when we are taking this, now the effect may be either by modifying the enzyme activity. So these drug molecule, they will modify the enzyme itself, or sometimes it may be that they are stimulating or suppressing certain receptor. So either they can modify enzyme's activity, or they can directly interact with the receptor. They can stimulate it or suppress it. Fine. Enzymes as drug target. Now, if we focus on this, that is how the drug molecules they are interacting with enzymes. So now the enzymes are as if the target, not the receptor. Just see these 2 points. They can modify enzyme activity or they can interact with receptor. So first we'll see enzymes as drug target. That means the drug molecules, now they are targeting enzyme. See, if we want to understand this, there are 2 points. The first point is we have to understand what is the catalytic action of enzyme. We know they are biological catalysts, but how they actually works. If we know that, then only we can understand how drug is modifying their activity. So first we have to understand how enzymes itself, it is doing this catalytic action. So for understanding the interaction between a drug molecule and an enzyme, first we have to understand how do enzymes catalyze the reaction. Okay. Now when enzymes are doing this, in their catalytic activity, enzymes perform 2 major functions. The first function of an enzyme is to hold the substrate for a chemical reaction in a suitable position. Okay. So see, here we have enzyme and this is substrate. Not all the substrate are meant for all the enzymes. There is obvious some specificity, specific substrate, specific enzyme, and that depends on the shape of this active site of the enzyme. So see how it is shaped. it is matching, it is complementary shapes, it is matching with it, and this is the active site of the enzyme. So the first function of an enzyme is to hold the substrate for a chemical reaction in a suitable position, and they do so through the active site of the enzyme. Now once it is properly fitted, now they can be attacked by the reagent effectively. That is, now the reaction is possible. The substrate is sitting properly on enzyme on its active site. Now the next step is possible. So this is substrate binding, and some reagent will interact with it. That is not shown here. It is a simple picture, that basic simple picture. Next, once the reaction is finished, the substrate can bind to the active enzyme. Okay, this point is also important. How this interaction that you are seeing here, how this interaction is possible? It may be through hydrogen bonding. It may vary depending on what type of groups are present. It may be hydrogen bonding, it may be ionic interaction, that is positive charge, negative charge, or it may be simple van der Waals force. So by any of these interaction, there may be this substrate can sit on the active site of enzyme. Now once the reaction is complete, now what will happen? There will be cleavage. See, already the substrate, it is converted to product, and the products, they will be released. So this is the first function of this, the picture that you are seeing here. Now the second function of an enzyme is to provide functional group which can attack the substrate and perform enzyme catalyzed reaction. So sometimes not just sitting on the active site, they can also provide some functional group which can attack the substrate. So this is another way by which also enzyme can interact with substrate. Okay, so these are the catalytic action of enzymes. Now we will see the second point under enzyme as drug target, which is drug enzyme interaction. Now the actual thing, how the drug molecules, they are targeting enzyme, what type of interaction it is doing. So drugs can inhibit the activity of the enzyme by binding to its active site, where originally substrate molecule should sit, but now substrate is not sitting, drug molecule is sitting. So there may be some that is drug, it is inhibiting the substrate to sit on its original position. So thus binding with the active site of enzyme, it inhibits enzyme catalyst reaction. So it may be that sometimes this enzyme catalyzed reaction is not desired. We have to inhibit it. So that is why the drug is now inhibiting in this way. The chemicals that inhibit the activity of an enzyme, they will be called enzyme inhibitor. Okay, so it is making some it is creating a situation so that original normal conditions cannot occur. Now drug inhibit this type of action of enzyme, that is, it is disturbing the enzyme activity, inhibit the attachment of substrate on the active site of enzyme in two different ways. Okay, so suppose there is no drug molecule. In the previous slide we have seen that substrate is interacting with enzyme and there is some products formation. But suppose if this is not desired, now the drug, it is doing this inhibition so that the normal process cannot occur between substrate and enzyme. But it can do so in two different ways. So one by one we will see. Among these two ways, the first one is competitive inhibitor, where there will be direct competition with the substrate molecule, because substrate should sit originally, not inhibitor. So inhibitor, the picture that you are seeing, you can see if this is the active site, which is V shape, it is properly matching with both substrate and inhibitor. So there will be some competition. Substrate should originally sit, but now the drug inhibitor is sitting. The substrate is rejected, right? So instead of getting enzyme substrate complex, now you are getting enzyme inhibitor complex. So in this way, inhibitor binds to the active site, so desired reaction is not possible because it is directly blocking the active site, and that is it is competitive inhibitor. This drug will compete with the substrate molecules by attaching themselves to the active site. Okay. So in this way, it is not allowing the substrate to attach itself to the enzyme. Obviously, the reaction is not possible. But in this case, one point you have to remember, if the concentration of substrate is very high, suppose you have taken drug, but compared to the number of drug molecules, if number of substrate molecules are very high, so obviously the chance that the substrate will sit on the active site, that is also very high. So if it is so that substrate is far greater, number of substrate is far greater than that of drug molecule, then this method may not work properly. Okay, it will be inhibited, but not complete inhibition is possible. So this is competitive inhibitor, inhibiting action of the drug, and the second one is non-competitive. Another name is allosteric. Now what is allosteric? We will also see that. In drug, this is the active site, that is fine, but see, this is another site which it is shown here in semi-circle shape. Not necessarily it has to be semi-circle, but just for our understanding, it is given in this way. So these drugs bind to a site which is not active site. So it is something other than active site, and that site is known as allosteric site. Fine. This drug binds to a site other than the active site, and as a result, now what happens? There is a conformational change to the active site. So it is true that the drug is interacting to the allosteric site, not to the active site. But because of this interaction somewhere else, not in the active site, now what will happen? There will be conformational change. So see, initially it is V shape, but now the shape is changed, right? And why it is so? It is because of this allosteric interaction. Fine. Now once this inhibitor, it is approaching this allosteric site, there is change in the conformational shape, and as a result, now what will happen? Because of this change, now the substrate, there is no such matching, so it will not sit here, it will not be able to recognize. As a result, the substrate cannot recognize the enzyme and won't attach itself to the active site. So this is also another kind of inhibition, right? Though in this case, unlike the previous situation, it is not sitting, drug molecule is not sitting on the active site, so there is no direct competition. It is doing its role somewhere else, not in the active site. So as if it is an indirect inhibition. Okay, and no reaction is possible because of the change of shape in the active site. In the previous case, it is directly sitting on the active site, no change in shape is observed, but now it is not directly sitting on the active site, but it is making some change, conformational change. So that is why reaction is not possible, because substrate will not be able to recognize the active site. Now after finishing all this, just see, suppose if the bond formed between this enzyme and inhibitor, because inhibitor is interacting with enzyme, so if somehow this bond is very strong covalent bond and cannot be broken easily, so that is also a problem, because we need, for normal function of our body, we need the enzyme in its normal form back. But if this interaction is so high, then it will be blocked permanently, but that is not desired. So in that case, what happens? The body degrades this enzyme inhibitor complex if it is not naturally broken, and as a result, we can get back the enzyme, or if it is not possible, then the body can synthesize new enzyme also, because the body is so intelligent. Okay, now problem based on this. Statement about enzyme inhibitor drugs, the discussion that we have just finished. 4 statements are given. We have to choose correct answer from the options. In each option, there is 2 or 3 statements present. The first statement, there are competitive, non-competitive inhibitor drugs. Now this is obviously true. 2 types of mechanism we have seen. So this is true. Now A is present in all the options. So just based on this, you cannot eliminate any option. Now the next one, this can bind at the active site and allosteric site. That means what? We have seen that it is when competitive inhibitor, it is sitting on active site. When it is non-competitive, indirect, then it is sitting on allosteric site. So they can bind at the active site as well as allosteric site. This is also true. Now once A, B both are true, these two not possible. The third point, competitive drugs are allosteric site blocking drugs. Non-competitive drugs are active site, but actually the reverse is true. Competitive drug, they directly sit on the active site. Non-competitive drug, they sit on allosteric site. So both these are wrong. If you interchange them, they will be correct. So both C and D false. And we have to choose correct statement, right? So A and B correct only. Option 3. So drug can inhibit the attachment of substrate on active site of enzyme in two ways. Competitive inhibitor related to active site, non-competitive or indirect inhibitor, they will be related to allosteric site. Now we have finished how drug they are targeting enzyme. Next topic is receptor as drug target. Enzyme as drug target, finished. Receptors as drug target. Now the drug molecules, they interact with receptor. Many drugs, they exert their physiological effect binding to a specific molecule which is known as receptor. So what is receptor? Just we have some basic idea. So receptors are mainly composed of protein molecules, fine, just like enzyme. They are integrated, or you can say embedded in the cell membrane in such a way. They are embedded in a special way. They are embedded in such a way that their active site is exposed outside the region of the cell membrane. It is not towards inside the cell. Outside the cell, it will be exposed. And the other part, rest of the part, except active site, rest of the part of receptor, they will be present inside the membrane. So this is the design of receptor molecules. The outer part of the receptor, they will only interact with the targeted drug. That is, from receptor point of view, they will also have some targeted drug. If I say from drug point of view, there will be some targeted receptor. So both are basically targeting each other. So you can see from this picture, this is cell membrane. Okay, this is the interior of cell and this is the outer region. Now if you look at the receptor molecule, this is actually the active site. Now the active site, as if they are protruding outside the cell membrane, and rest of the part, they are inside the cell. Fine. So this is how receptors are integrated in the cell membrane. These receptors, now we will have some idea what is their role before going to the actual interaction with drug. So these receptors help us communicate via certain specific chemicals, which are known as chemical messengers. So they work through these chemical messenger molecule. These messenger molecules carry message. That is why their names are messenger. They carry message to cell, from cell to cell, by binding to the active site of this receptor. Fine. There are a large number of different receptors in the body that interact with different chemical messengers. So not just any chemical messenger is meant for any receptor. There is some specificity. For a particular receptor, there is a particular chemical messenger. Just same thing that you have seen in case of enzyme. For a specific enzyme, there is a specific substrate. And based on how the active site, the complementary shape must be present in the substrate. Same thing. Same thing here also. Receptors show selectivity for one chemical messenger over the other because their binding sites have different shape. As I have said, the same thing you have observed in case of enzyme substrate. So here also, because of different shape, different structure, and amino acid composition. Because after all, they are protein molecules. And protein means what? It is a polymer. If you consider as polymer, that the individual monomer units are amino acid. So depending on what type of amino acid sequence is present, amino acid composition, what are the groups present in the side chain of amino acid. Depending on all these things, their binding sites will be different, and those chemical messenger which will have complementary site, they can only bind. Fine. So in this way receptor works. So if you look at this molecule, it will be clear how the receptor works with chemical messenger. So cell membrane, as you can see, this is the outer surface of the cell membrane, this is the interior of the cell, and this blue colored picture that you are seeing, this is actually the receptor, and the pink one, that is chemical messenger. So the binding site, as if it is, these two are complementary, so it is fitting properly, induced fit. Now there will be some message, very simple picture. After it is sitting on this site of receptor, and once the message is given already, its work is finished, now it will go back to its original position. Okay, so in this is the picture from which you can understand how receptor, they interact with chemical messenger molecules and some cell signaling is possible. Now drug molecule will come into the picture. They can target receptor molecules again in two ways. The first way is, there are some drugs that binds to the receptor site to inhibit its natural function and block the communication of the message. So they will bind to the receptor site and block the communication. They are known as antagonists. Here we have example, adenosine. It binds to the adenosine receptor, and its message, what message it gives? It induces sleepiness. Now the drug that you are taking, that is the antagonist drug, suppose it is caffeine. This antagonist drug caffeine, now what happens? Instead of adenosine, now it binds to the same receptor where originally adenosine should sit. But now adenosine is not sitting, caffeine is sitting, and it is making this, blocking this communication, which is actually, that is, if it is original, suppose no caffeine is there, then original message is inducing sleepiness. But now adenosine, it cannot work because of presence of caffeine. So caffeine is making it inactive. As a result, there is reduction of sleepiness. That means there will be wakefulness. So in this way, caffeine works. When we take, eat, consume caffeine, there is reduction in sleepiness, right? So this is a way by which drug molecule can interact with receptor, and they are known as antagonist. The second way is agonist. In this case, there are some drugs which mimic the natural messenger. Now they are not directly sitting, they are mimicking the messenger by switching on the receptor. Okay, try to understand the difference. In the previous case, it is directly sitting, directly interacting with the receptor site, but now they are mimicking the natural messenger. So there may be some similar type of grouping, so that is why it can mimicking or copy basically. And they are required, this type of drugs, that is agonists, are required when there is a lack of chemical messenger. So suppose some chemical messenger, it has a particular role. For example, you can consider this example, morphine. Its role is to kill pain killing, fine. So it binds to the receptor which is known as opioid. It suppresses the neurotransmitter that causes pain, fine. So in this way, it is mimicking the natural messenger, and by switching on the receptor, fine. So morphine is the agonist drug which is used as a painkiller. Now it is binding to the opioid receptor, suppressing the neurotransmitter that causes pain. So when it is mimicking, just remember this, it will be called agonist. And when it is not, that is not by mimicking, it's doing, playing its role, then it is antagonist, fine. Drugs which bind the receptor site and inhibits its natural function. So here we have options: chemical messenger, agonist, antagonist, neurotransmitter. Now chemical messenger, this is obviously not true. Then neurotransmitter, there, that is also same thing. That is also one type of messenger. But you have to choose from these two. Now it is not saying anything that it is mimicking. It is binding to the receptor site. So correct option will be antagonist. Some drugs bind to a site other than the active site of enzyme. Remember how the drugs, it interacts with enzyme in two ways. Sometimes it interacts directly with the active site. Sometimes some other place which is not the active site. And this other place, that site is known as allostetic substrate non-active competitive. Now this is correct answer. Remember, the other site is basically known as allostetic site. There is no such term we have used which is substrate site. Okay, there is no such term exists at all. Then active site, this term is, it exists, it is important, but there is no such term as non-active. Okay, competitive site, no, all these terms are actually none of them exist. Okay, so correct option is allostetic site. Therapeutic action of different classes of drugs. So what type of therapy they are, that is, what type of therapy we are getting from this drug? We can do this classification, and here you can see total 7 classification. Some class names under which also there will be some sub classification, subdivision will also be there. So one by one we will discuss them. Antacid, as the name suggests, it is something which is inhibiting the acid production. So that is why the name antacid. We know that in stomach, because of digestion, there is natural production of HCl acid, hydrochloric acid. But if it is excess, that is more than required, there will be obviously some irritation and pain. Even in some cases, because of this excess acid production, ulcer may also happen. Okay, so this is really a problem. So as a definition of antacid, we can say the substance which neutralize the excess acid and raise the pH, because when it is low pH, that means it is acidic condition. When it is high pH, more than 7, we say it is basic condition. That means our purpose here to increase the pH from low to high. That means to move from acidic condition to basic condition. So the substance which neutralizes this excess acid and raise the pH to an appropriate level in stomach. Appropriate level means the level which should be there, which is desired level. These substances are known as antacids. So for example, sodium bicarbonate, magnesium hydroxide, which is also known as milk of magnesium. It is insoluble in water, not soluble. Aluminium hydroxide, etc. That means metal hydroxide or sodium bicarbonate. We know this base, it is inorganic base. It's commonly used in laboratory. So they can also be used as antacid. Okay, but the problem is they can treat only the symptom but not the actual cause. So they are, as we know, they are basic molecule, metal hydroxide or sodium bicarbonate. So it is simple acid base reaction, and as a result pH will gradually increase. So they are basically just controlling the symptom, but for what reason this excess HCl is actually produced, that cause is still there. So if we use this type of antacid, sodium bicarbonate, MgOH2, then complete relief for long time it is not possible. It can give us some temporary relief. Fine. So we have to know the actual cause. What is the actual cause? The secretion of pepsin, which is basically one of the enzymes, and it helps in the digestion of protein. It breaks peptide bond. That is why the name pepsin. And also the secretion of HCl. Mainly we are interested in this molecule. Is stimulated by histamine. What is histamine? This is the structure. There is imidazole ring, which is heterocycle 5-membered ring with 3 carbon, 2 nitrogen, and then there is a chain CH2, CH2, NH2. This is the structure of histamine. The secretion of HCl, it is controlled by this histamine. Now, there is a drug which is cimetidine. It is far better than this previously mentioned drug. Now, this drug, cimetidine, it is designed in such a way so that it can stop the interaction of histamine with the receptor, which receptor that are present in the stomach one. So what happens? Suppose there is no drug we have taken, whether it is NaCO3 or cimetidine, whatever. So what we will do, that as histamine is controlling this secretion, so when they will interact with receptor, there will be some cell, that is some message will be exchanged. As a result, there will be production of more HCl. But if somehow we are taking this cimetidine, now they are designed in such a way they can stop this interaction. And when this interaction is stopped, now the actual cause we are hitting. So because of this, now less acid will be produced. And the problem of this hyperacidity, now it can be checked for long time. Not just cimetidine, there are some other examples also. Ranitidine, mostly the brand name is Antacids, omeprazole, lansoprazole, laviprazole, etc. So here we have picture of cimetidine. Exact picture, it is really not possible to remember. Just try to remember the basic structure. When I'm saying basic structure, I'm trying to say basic structure. Here you can see this ring heterocycle is present, and there is some chain. In case of other one also, ranitidine, there is no imidazole, but there is furan ring, and there is 2 chains. And lansoprazole, omeprazole. Now in these 2 cases, you can find some similarity. So now this imidazole ring, it is also fused with benzene ring. It is known as Bain's imidazole. Okay, so all these are examples of antacid. Fine. Sometimes structure related questions are also there. We'll obviously discuss. So then that is why to have some minimum idea about the structure is also important. Fine. So we are at the end of the session. We'll complete the rest of the discussion in the next class. Thank you for listening.